Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.7, Tonicity and Osmoregulation. This is one of the more difficult concepts within AP Biology because of the equations involved, but also the numerous ways to say the same exact thing, all three of which could appear on the AP exam. But today we're going to be focusing on hypertonic, hypertonic, or isotonic environments and the movement of water between these environments. We'll also be looking at the water potential and solute potential equations. Suggested skill is representing and describing data for A. Construct a graph to represent the data, including XY graphs, bar, histogram, line, log scale, dual Y, scatter plot, box and whisker plot, and pie chart. Graph should include the following components: type of graph appropriate for the data, axis labeling, including appropriate units and legends, scaling, accurately plotted data, including error bars when appropriate, and trend line when appropriate. So this is also a crucial skill because you want to be able to graph it as well. Hypertonic, hypotonic, and isotonic environments. So, like I said, osmosis is the diffusion of water across the cell membrane. This is going to require an aquaporin, which is a membrane protein. And if you recall, that means this is going to use facilitated diffusion, membrane protein to travel across the cell membrane. Now, water in the cell is going to have a relationship with its surrounding environment. The amount of water is always going to be proportional to the amount of solute, or stuff, molecules in the cell. So if we have this, you could say, U-tube of a beaker that has a semi-permeable membrane that only water can pass, and there's going to be a lot of solute here on this side and very little on this side. Even if water starts out at an equal level on either side of the tube, eventually there's going to be way more water on this side than this side, because the water is going to be proportional to the amount of solute in the side of the tube. The water is always going to follow the amount of solute. Now, there are three solutions that represent this relationship between water and its environment. First of all, an isotonic solution, there's not going to be any net movement. There's not going to be change in the amount of water. Water will move in and out of the cell, but it's not going to be a noticeable change because there are going to be equal amounts of solute both in and outside of the cell. In a hypertonic solution, water is going to move out of the cell because there's more solute in the environment. Water is going to follow the solute, and this could cause cell shrinkage. Lastly, in a hypotonic solution, water is going to move into the cell because there's less solute in the environment and more in the cell. Again, water is going to follow the solute. It wants to establish a proportional relationship. So now let's get into the math. And always on the AP exam, they will give you the formulas, but you still need to know how to use them and how to properly plug in numbers. So in case you do get a question relating to water or solute potential, you'll be able to solve it. So water is going to move from an area of high water potential to low water potential. Now, what is water potential? It's the ability of molecules to flow freely. So high water potential, lots of molecules are able to flow freely. That means there's not much solute. And it's going to move to a low water potential where there is more solute because water always follows the solute. So high water potential to low water potential. It'll also move from low osmolarity to high osmolarity, from small amount of solute to a higher amount of solute. And lastly, from a lower solute concentration to a higher solute concentration. So water is always going to move towards the solute concentrate, to the higher solute concentration. And this is inverse to the water potential. And solute potential is the amount to which water potential is lowered by solvent. So basically, with a high solute potential, you're going to have a lower water potential because there's going to be more solute. And lastly, water potential is equal to the solute potential plus the pressure potential. Now, in most cases, the pressure potential is going to be zero because you're going to be dealing with open containers or canisters or test tubes, generally when they describe this experiment. If not, then they will tell you what the pressure potential is. So now all you have to do to find your water potential is to be able to find the solute potential. And for this, they will again give you the formula, but it's negative ICRT. So what does I stand for? It's the ionization constant. How many ions are going to be in our solute? C is the molar concentration, moles per liter, and remember, moles is just a way we measure molecules. Next, there's a pressure constant, 0.0831 is always going to be your pressure constant. And lastly, you need your temperature in Kelvin, which means you take your degrees in Celsius and you add 273. So over here on the right, I've got an example. We've got one molecule of sucrose with a molarity of 0.42 moles per liter at 25 degrees Celsius. And we're going to find first our solute potential and then our water potential. So solute potential is going to be negative I, ionization constant. How many ions within the molecule of sucrose? There are none, so our ionization constant is 1. This molecule of sucrose is not going to split up into multiple ions if you place it within water. Next up, you have our molar concentration of 0.42. Then our pressure constant, 0.0831. And lastly, our temperature in Kelvin, which is going to be 273 plus degrees in Celsius, which is 25 degrees in Celsius. That gets us to 298 degrees in Kelvin. And after we multiply all of this up, we get a solute potential of negative 10.4. And obviously adding this to our pressure potential of 0, we get our total water potential is going to be negative 10.4. So there's not a very high chance that you will see a question on this on the AP exam, but it's still good to know the different terminology, how water is going to move always towards more solute, and how to find both water potential and solute potential. So let's talk about tonicity in plant versus animal cells. In an isotonic animal cell, that's a normal concentration. We don't want water moving in or out of the cell in a net movement, just dynamic equilibrium. It's movement, but it's not changing significantly. Now, an isotonic plant cell is known as flaccid because plant cells like pressure. So they want water to be moving into the cell a little bit, or else they're going to collapse a little bit. So a hypotonic animal cell, this is where water is moving into the cell because there's more solute there, is known as lysed, and it's going to cause the cell to explode. We go kaboom. In the hypotonic plant cell, this is known as turgid because it's a normal amount of pressure. This is what the plant cell likes. It likes to have pressure pushing into the wall. Now, lastly, hypertonic animal cell, water is leaving the cell. It's going to become shriveled. And a hypertonic plant cell is going to become what's known as plasmolyzed. As you can see, the membrane within the plant cell is going to shrink, but the plant cell wall remains the exact same. Because this plant cell wall is not going to respond to these changes in water. It's a sturdy structure that's going to stay in place whether water has moved out or into the cell. However, the membrane is what's going to expand or contract. And because of this, plant cells can also not burst with too much water in a hypotonic solution. Lastly, contractile vacuoles exist in some animal cells, such as the paramecium, and these can pump water out of the cell in case they're placed in a hypotonic environment and lots of water is moving into the cell. A contractile vacuole is able to pump this water out to keep the cell at an isotonic solution level. This was 2.8 tonicity and osmoregulation. You want to know about osmosis, that diffusion of water through the aquaporin based on whether the solution is going to be hypertonic, hypotonic, or isotonic. Remember, water is always following the solute. And then lastly, you want to know how to calculate water and solute potential. Next up, we're going to do a quick review of all of the different kinds of membrane transport.